This is a review video for Chapter 7 and Chapter 8. Chapter 7 is Cellular Respiration and Chapter 8 is Photosynthesis. So on this first slide we have a picture of, you might already know what it is. It's a monarch caterpillar, which will soon be a butterfly. And you may already know that they are almost endangered. Their numbers are extremely limited. And you might also know that they're toxic. They have these kind of warning colors, this yellow and black and white. And so really the only reason that they're toxic is because when they're a caterpillar, they eat the milkweed, and the milkweed plant is toxic. So it's not until they actually eat this plant that they become toxic. So this is a warning to birds. Don't eat me, I am toxic. And I'm not sure that it really kills the bird, but I know it is toxic for them. So, but anyways, it's titled How Cells Harvest Energy. And so we're gonna go through how, once you eat your food, how is that converted to usable energy on the cellular level. And so we start off with autotroph versus heterotroph. And so like for us, we cannot make our own food. We do need to go out and eat food, and so that's why we're considered to be a heterotroph, right? Organisms that can't make their own food. Something that can make their own food are things like plants. Plants are able to absorb the sun and then convert that into a sugar, and then that sugar is then converted into a usable energy on the cellular level. So that's the difference between an autotroph and a heterotroph. And so some of the things that help along the way are what we call electron acceptors. And then also some distinguishing features between aerobic and anaerobic respiration is that one is using oxygen and one does not use oxygen. So cells that are eukaryotic and much larger typically need more energy, and so they're going to be using primarily what we call anaerobic respiration. But they are able to use anaerobic, right, when there is no oxygen present. Cells that are much smaller, like bacteria cells that are prokaryotic, some of them are able to live in places where there's no oxygen. Remember, on the earth, there wasn't always oxygen, but there was still life. So the bacteria that was living on Earth without oxygen, how were they able to make energy? Well, they were able to do it through this process called anaerobic respiration. And then the last one is fermentation, which is also when there is no oxygen present that you can ferment. And so maybe you've heard that word before, like fermented foods like beer, yogurt, cheese. That really is what this is, fermentation. So again, for aerobic respiration, this is the one that does need oxygen. And so the chemical equation is the C6H12O6. So that big molecule is your glucose sugar. So your sugar plus your oxygen is going to yield CO2, water, and energy. And so when you see the term aerobic respiration or anaerobic respiration, and then you hear cellular respiration, those are all the same thing. This is just converting energy into usable energy that the cell can then use. And so what we mean by energy, that could be like in the form of ATP that then gets used by the cell. When we talk about free energy, there is actually like a quantitative number attached to it. So free energy is negative 686 kilocal per mole of glucose. And then at the bottom it says, this large amount of energy must be released in small steps rather than all at once. So the ATP, I'm sure you've heard of this term before, but what really is it? So it's called adenosine triphosphates. And so that means that there's 3 phosphate groups, and so the energy really doesn't come from just the molecule like that, but when those bonds are broken, energy is released, and then that is then used technically as fuel for cells. And so there's 2 ways that you can actually make this, and so here's a picture of one. And so this is called the substrate level phosphorylation. So when you see this word phosphorylation, that means that you are adding a phosphate group. So here in this first picture, I've got the large enzyme, which is a protein. And so PDP comes and it has a phosphate group and it needs to attach it to this over here. And so notice right here it says ADP, so that's diphosphate because there's two. So for phosphorylation, you're just going to add a phosphate group, and here this is using an enzyme to help with this process. And then N comes out your adenosine and then your 3 phosphate groups on the other side, and then some pyruvate, which is the product of glycolysis. So when we're talking about this oxidation of glucose, that's just kind of like how we're breaking it down. And so it goes into these steps. And the first one is glycolysis, and then pyruvate oxidation, the citric acid cycle, which is also known as the Krebs cycle. So if you ever see Krebs cycle or citric acid cycle, just know that they're the same. And then the last one is the electron transport chain. Here's kind of a brief overview of what it looks like. So you might already know that the mitochondria is the powerhouse of the cell. So cells that have a mitochondria are going to be your eukaryotic cells. So cells that are not eukaryotic, they don't do this. They don't have a mitochondria, so they cannot do aerobic respiration. So it's not unless you have the presence of the mitochondria and that you have oxygen that you can do this process, the aerobic respiration. So you have your glucose, and it's broken down in that first step, which is glycolysis. And the important thing that you need to know about this is that it happens outside of the mitochondria. So glycolysis happens in the cytoplasm. And so bacteria, they don't have a mitochondria, but they do have a cytoplasm. So bacteria can do glycolysis. So all cells have cytoplasm. So all cells are able to do glycolysis. And so that's something that you do need to remember. So glycolysis does happen in the cytoplasm, and then you have pyruvate getting broken down into acetyl-CoA. It goes into the citric acid cycle, which is also known as the Krebs cycle. And then lastly, it goes into the electron transport chain, and then chemiosmosis, ATP synthase, which is like a proton gradient. So, but also you have the outlets of each, like you could see it here on the left, like the product of the acetyl-CoA is that it produces NADH, and then all of these do come down and they help at the end with the electron transport chain. Another thing too is the location. So like the citric acid cycle or the Krebs cycle, it happens in the matrix of the mitochondria, and then the electron transport chain happens on the inner membrane. Typically you'll see that it happens in what we call the cristae, which is these folds. When you see these folds in the inner membrane, that's where you have a lot of this action going on. So just kind of keep those parts in mind, the cytoplasm, the matrix, the inner membrane or the cristae membrane. Just try to keep those in good memory. So for glycolysis, for each glucose, it's going to yield 2 pyruvates, and again this happens in the cytoplasm, and it also makes 2 ATPs by the substrate level phosphorylation, which is the one that we showed you earlier, that the enzyme helps make that one. And then also 2 NADH produced by reducing the NAD+. And so you'll see that NADH is very important, right? It says here for glycolysis to continue, NADH must be recycled to NAD by either. So it's the same thing that we talked about earlier, like how ADP turns into ATP. Same process for this. NADH and NAD+ are just in this kind of cycle where they're going from NADH to NAD+, NADH, NAD+. Same for ADP, ATP. It's just this cycle that's going on. What we mentioned earlier for the pyruvate, it does depend on how much oxygen is available. So if there is some present, then it could get further oxidized and then it enters the citric acid cycle. If it doesn't, then it is not able to go through, well, let's go back to this picture. So if there is no oxygen present, what happens to the pyruvate? Because it can't enter inside of the mitochondria, what's going to happen is it's going to go through fermentation. So let me go back to that slide. So if it doesn't have any oxygen present, it cannot continue the process, and it then ferments, and then you're going to get a lot less ATP at the end, and it might show you at the end how much of a difference in ATP, but if it doesn't, I'll just say it right now. For fermentation, you're only going to get a yield of 2 ATPs versus aerobic respiration. It depends. Some books will say like 32 to 36 ATPs, so it's a pretty drastic difference. But keep in mind, the anaerobic respiration does produce less ATP, but that's okay. Like bacteria cells are much smaller, and so there isn't really this need to make excess ATP. So it's just beneficial in some ways. Here's a picture of the electron transport chain. We talked about that it's on the membrane, on the inner membrane specifically, and so you just kind of have this proton gradient. So these hydrogen ions, and then you have the electrons just passing through, and then ATP synthesis here on the right, and it rotates and it rotates, and this is how it's able to generate all those ATPs. So let me see, is there anything on here? Yeah, each complex in the chain operates as a proton pump, driving protons to the inner membrane space. Electrons move from protein complex to protein complex. So these blue things are what we call the proteins, and so it's just jumping from one to one to one until it makes all of that ATP. And then here they just kind of show you, I guess, a zoomed in picture, but it shows you the movement of these electrons, how they're passed through, and how eventually you get all these ATPs. So here, notice how it says 28 ATPs. That's going to be a total coming out of this ATP synthase. And so this is the yield that this textbook says. So if it is a bacteria and there is oxygen present, that it could make a total of 32. For eukaryotes, it's around 30. So it depends on the type of cell, really, is what it is. So for anaerobic, we said that there are some organisms that are able to survive without oxygen, and so here's like a few examples, like this one, the sulfur prokaryotes and the methanogens. These are just organisms, bacteria that have able to survive even before oxygen, and so they're able to do anaerobic respiration, and they're successful. Nothing wrong with that. The fermentation we talked about a little bit. Common examples are like in our foods, and so we call this ethanol fermentation, and that happens in yeast. So like when you bake bread or whatever, you put yeast, warm water, sugar, and then flour, and then it expands. And so what it is, it's producing some ethanol, which is an alcohol. And so also that's how you make beer, similar but not in the exact same way. The other thing is the lactic acid. Maybe you have heard of this for athletes. Athletes that exercise rigorously, or you don't have to be an athlete, just anybody that exercises rigorously. If you're like running, it's really hard for you to take in a lot of oxygen, and so the next day your muscles feel really sore, and that's why, because there wasn't any oxygen present, and that it's going to produce this lactic acid, and that's what makes you feel sore. And so that's why it says it here occurs in animal cells, especially the muscles. And so that's a wrap up of cellular respiration. And the next one is photosynthesis. We've got a really gorgeous sunflower here. We already said that plants are autotrophic. They can make their own food, and so they do that by absorbing light and converting that into a sugar. And so here's the formula for that. It has to intake CO2, it needs water, it needs light, and then it yields a glucose, some water, and some oxygen. So, but what's interesting about plants is that they do both. They don't only do photosynthesis. They do photosynthesis and then they do cellular respiration. Because notice, so with this equation, this is showing you that they make the glucose But then it can't just use glucose like that. It has to break it down into usable energy, which is ATP. So after it makes the glucose, then the sugar does go, it gets broken down in the cytoplasm and then in the mitochondria. So just keep that in mind that plants do both. They do both photosynthesis and cellular respiration simultaneously. It doesn't have to be a plant. So on this slide, they mentioned that it could be cyanobacteria, which is a photosynthetic bacteria. Some algae and land plants have chloroplasts. Those are things that can do photosynthesis. There are two stages that we talk about, light dependent and light independent, meaning that one needs light and one doesn't need light. And so we'll kind of go and touch those a little bit more. And so one of them uses the ATP that was made in the light dependent, which is this one. So light independent, it doesn't need light, but it uses ATP. And so where did it get that ATP from? It got it from the light dependent reactions. And so these are happening simultaneously to fuel each other. The chloroplast is an organelle that's found within the plant. And so what's interesting about the chloroplast and the mitochondria too is that both of them, they have their own DNA, they have their own membranes, and so that's why we say that they used to be their own bacterial cell, and we call that the endosymbiotic theory because of those traits. But some of the anatomy of the chloroplast is that they have their own membrane, and they also have a thylakoid membrane, which is an internal membrane, a grana, which is a stack, and I think they have, let me see if we can get you a picture. There we go. So here in this picture, you can see it a little bit better. There is an inner membrane and outer membrane, the thylakoids, and a grana. It's not labeled here, but a grana would be a stack of these. The stroma lamellae is going to be this line that connects these stacks. And is there anything else on there that they have labeled for you? No, but so that's also chloroplasts are relatively large. If you look at this picture, like there's three here, and look how large they are. They're actually even bigger than the mitochondria over there. So just keep in mind that they're actually really large structure, probably larger than you may think. And so because we just went over cell respiration, notice how it looks kind of similar, that it has its own membrane. And there's, I guess, a lot of activity going on, but they have their own membrane, and then they have what we call photosystems. So there's one here, this photosystem, and all that is a cluster of pigments. So inside of this blue thing, there's all these green dots, and so those are just individual pigments. And so the cluster of pigments we call a photosystem. And so the sunlight is going to hit those pigments, the pigments get excited, and then that's when the reaction actually occurs. And so here we talk about the pigments. Light is a form of energy, and we call those like photons. So whenever you see photo, that root word photo, that means light. So a photon is a particle of light, and so that's what I guess triggers this whole process. So kind of interesting in that sense. As far as on the scale of how these photons get activated, it has to be with visible light. And so the spectrum for that is going to be the 400 to 740 nanometers, and that's what we call visible light, between 400 and 740 nanometers. This is really cool because on this graph, you kind of see when the pigments are active. So like chlorophyll A, it's active here and here. Chlorophyll B, I'm not sure if I can mute that. Chlorophyll B is active between it peaks at 450 and it kind of peaks at 600. And so you can kind of see where they peak, but notice here in the green, they don't peak at all. So that's interesting because plants are green, but they don't absorb green light. So when you see a green plant, that's because it doesn't absorb the green pigment, it reflects it. And so that's really interesting to remember. Some of the pigments, we talked about chlorophyll. The other one is the carotenoids. Carotenoids are going to be they're like a yellow, red, orange kind of color. So those are actually cool as well. There's two types of chlorophylls, chlorophyll A and chlorophyll B. Chlorophyll A is the main pigment in plants. They absorb violet, blue, and red light. Chlorophyll B is a secondary pigment or accessory pigment, and they absorb anything that chlorophyll A doesn't. And so when you go back to the graph that we were looking at, notice how they don't overlap. Chlorophyll A is the solid line and chlorophyll B is the dotted line. So that's how they're differentiated. As far as chlorophyll as a structure, it has this hydrocarbon tail, so it's considered to be non-polar. And actually, if you've ever gotten a grass stain, like where you slid on grass outside, it's very hard to get that out of your shirt. And then this is kind of why, because it's a non-polar, so you can't get it off with water. This is what a photosystem looks like. We already established that it's a cluster of pigments, specifically chlorophyll, and so when the photon, which is light, hits it, that's when it gets activated, and then you have all of these reactions occurring. And so you'll have like electrons and electron acceptors. And so that's really it for as far as that. For light dependent, remember this requires light. So the photon, it's captured by the pigment. Energy is transferred to the reaction center. An excited electron is transferred to an acceptor molecule, and then electron transport, which is similar to what we talked about earlier, like with the mitochondria, in their electron transport chain. Here it's similar, but it's not exactly the same. Electrons move through carriers to reduce NAD plus to NADPH. And then chemiosmosis is when it makes the ATP. The two photosystems, or I'm not sure if we spoke of them, but we did speak about photosystems. And so in photosynthesis, there are two primary photosystems, photosystem 1 and photosystem 2. So these are just, it's like a cluster of pigments, part 1, and a cluster of pigments, part 2. And so that's really all that you kind of need to know as far as how they're different. So here we've got the membrane, and then this is very similar to what you saw a little while ago on the mitochondria. You have proteins and the photosystems here on the membrane, and then look, you've got the ATP synthase again, just like you did in the mitochondria, and this is where it's producing the ATP. So again, you have this kind of like flow of energy. You see the electrons passing through hydrogen ions as well, very, very similar to what you saw earlier. And so this is how it's able to make the ATP. But check out how I've got photosystem 2 getting hit by the light energy. It gets excited, it passes the electrons, and then it goes to photosystem 1, same thing, light energy hits it, passes electrons, and then that's how you're able to make your ATP. So this is the last slide. It just kind of shows you the similarities and also a comparison between the two. So one, for photosynthesis, this does need light, and the other one doesn't need light. Both do make ATP. The difference, though, is that the chloroplast will make glucose, and the mitochondria is breaking down the glucose to make ATP. So there's a lot of overlap, but there are definitely some differences as well. And so hopefully this was helpful, and thank you guys for watching.